Today we're going to talk about the basic structure of DNA. So, there we go. So, the structure of DNA was first figured out by these two guys, James Watson and Francis Crick. It wasn't actually discovered until the 50s.
What's that one guy's name?
Crick.
Oh,
C-R-I-C. Okay, so if you remember yesterday, Friday when we stopped talking, scientists figured out that DNA was probably the thing that coded for your genes. The thing that Mendel was talking about when he was looking at alleles and how they passed on and all that kind of stuff. And so there was a big push to try to figure out how DNA worked and what its actual structure was. Okay, now before these guys came along, there was a lot of things that had been figured out. For one, they had figured out that DNA was made up of certain types of organic molecules called nucleic acids. And we've studied nucleic acids before. Does anybody remember what a nucleic acid is made up of? There's 3 parts. Proteins are a whole different organic molecule. DNA is made up of nucleic acids. So, oh, you guys just throwing words out. All right. For one, you have a phosphate.
Oh, something diphosphate.
Okay, and then you also have a sugar, and then you have a nitrogenous base. So we've looked at that before. And remember, ATP is a type of nucleic acid. Specific type of nucleic acid is just ATP has 3 phosphate groups instead of just 1. So they figured out that DNA was made up of nucleic acids, but what these guys were working on was trying to figure out how those nucleic acids went together and what the shape of the DNA actually was. They were able to extract DNA, but they didn't really know what the shape was. There was some evidence towards it being helical, meaning it kind of wound around, but they didn't know if it was a single helix or double helix, or some people had hypothesized that it might be a triple helix. And so that's what they were working on. They were the first ones to kind of figure it out. But they did it in sort of an underhanded way. These guys never did any actual physical work with DNA. They came up with the structure of it, but it was all based on other people's data. Now, most of that stuff was stuff that was published, and that's how science works. You publish your works, other people read it, and that makes them think of something. And so there's a lot of people that figure things out just by looking at what other people have done. But the underhanded part was they were talking to a guy that worked at a different college than them named Maurice Wilkins. And Wilkins was working with another scientist on what's called X-ray crystallography. So basically what they do is they take certain things and they crystallize them in a certain way, sometimes by freezing it, sometimes by reacting with chemicals, and then they shoot X-rays through it. And the X-rays go through the crystal, and then they hit a piece of photographic film behind, and the pattern that emerges they can use to figure out a few things about it. So they were working on X-ray crystallography of the DNA. They take chunks of DNA, they crystallize them, they shoot X-rays through and see how they turned out. But he wasn't the main guy that was working on it. The main person working on it was a woman by the name of Rosalind Franklin. And Franklin was the one that was doing the actual work. So this is an actual photograph plate taken by X-ray crystallography. And to us, it just looks like a bunch of just different smudges, but based on all the previous work that she'd done before, she could figure out certain things about the structure and the components of the DNA molecule by the way that this pattern of smudges showed up on her plate. But before she published her results, Wilkins let Watson and Crick take a look at her notebook without her knowledge. So kind of behind her back, they all went through her work and looked at it. And when Watson was kind of the genius part of the team, when he saw this, it all clicked into place. And he goes, oh, I know exactly how it goes together. And they went back and they built their DNA model. We go back to their picture. This is the actual model that they built. So each little peg here stands for a different atomic bond. So they've got all their carbons and hydrogens and everything. But you can see it's kind of a helical structure, kind of spirals up as it goes around. So based on that, they were able to figure out the double helix pattern. For their discovery, they were awarded a Nobel Prize in 61 or 62. I don't know, one of those years. Both Watson and Crick won it, and also Maurice Wilkins. Rosalind Franklin did not win a Nobel Prize. Unfortunately, by that time, she had died. She had gotten ovarian cancer at the age of about 36 or 37, and passed away before they ever gave out the Nobel Prizes. Otherwise, I don't know. It's hard to say because they only give out Nobel Prizes to 3 people at a time. So they would have been in a kind of conundrum. Who are they going to give it to? She's the one that really did all the work. Wilkins almost didn't really do anything. He just kind of showed them. He did help write their paper, but kind of actually saved the Nobel Committee from making a hard decision there. But if she had died, they don't give out Nobel Prizes to people after their deaths. And they figured that the ovarian cancer was more than likely caused by the fact that she was using X-rays and never really protected herself. Nowadays, when you go to the dentist, when they take an X-ray, they put that lead that you put on. You're not soaking up extra X-rays. But she was taking hundreds of X-rays for this stuff. Didn't really protect herself. So they figured that the ovarian cancer was likely caused by her overexposure to X-rays. Because of that. So unfortunately, she never really got to share in the glory of the finding of the structure of DNA. But from what Franklin was able to show and what Watson and Crick put together, we now know that the shape of the DNA molecule is a double helix. Oops. Should spell helix with an actual L. So you've got 2 kind of strands that wind around each other and are connected in the middle. And you can kind of think of it like a winding staircase, okay? These are the outside rails of the staircase, or the ladder, if you want to think about it like a twisted ladder. And in here are the rungs that you would climb up. Okay? I guess we should maybe back up and actually talk about what DNA stands for, although many of you maybe know that. Right? Deoxy, oops. Ribonucleic acid. DNA. And as we'll see, the deoxyribo part of that just describes the type of sugar that's going to be in the DNA molecule.
Isn't that acid?
Yes. I don't know what happened to my part of my D, sorry. Deoxyribonucleic acid. Okay, so we need to talk a little bit more about more precise structure and how these things fit together. And tomorrow we're going to talk about why there's a double helix, why there's 2 of them, and how this thing gets copied before a cell is able to split. Let's go back to that. So, what is this?
ATP. Nucleic acid.
Nucleic acid. Not ATP because there's only 1 phosphate group. ATP would have 3. That's why it's triphosphate. Okay? So this is a nucleic acid. So this would be 1 molecule of the DNA chain. So here's the sugar. In DNA's case, this is ribose. got your phosphate group, and then up here is your nitrogenous base. Now that can change. The shape of this part right here can change, and that's what we're going to have to talk about in a minute. But the basic setup of a DNA molecule is just a chain of all these different nucleic acids. This part right here might change a little bit, but the rest of it is all together. And the way it goes together is a phosphate group and sugar form the backbone. So this sugar will be bonded to another phosphate group, which is, got its sugar, and then it'll be bonded to another phosphate group, which has got its sugar. And that forms the actual backbone of the DNA molecule. So here we have separate separate nucleic acids with this phosphate sugar backbone. And that's what runs up the kind of outside rails of the DNA molecule. So as far as the chain, when we put a DNA molecule together, the chain, the nitrogenous bases don't really participate in the chain. It's all the sugar and phosphate groups that bond together to form the chain. Okay? So if we look here, we see sugar, phosphate group, sugar, phosphate group, sugar, phosphate group along one side of the DNA. And over here, same thing. Phosphate group, sugar, along the other side. Yes. Okay, and that leaves these nitrogenous bases. And that's kind of what we're going to focus on really for most of the rest of the chapter, because that's where the differences between the nucleic acids are. But it's also what causes DNA to be able to do all the stuff it does. To be able to tell your cells, hey, we need to do this, so that you end up with blue eyes or brown eyes. To be able to replicate, make another strand of DNA so that you can make new cells that have those instructions. To be able to pass on your genetics to your sons and daughters. All of that is caused by the fact that we have these nitrogenous bases and how they interact with each other. Okay? Now, as far as the nitrogenous bases go, there's 4 different kinds. Here's where we want to be. There's 2 kinds that have double ring structures. We call those purines. So, I mean, you don't have to draw these out, but I would just, maybe draw 2 circles and write double ring. And then there's 2 kinds that have single rings, and we call those pyrimidines. You can see that in all 4 cases, we have molecules that are made up of rings. They have nitrogen in them somewhere, and then some carbon and hydrogen. Some of them have oxygen, but as you can see, one of them on the end does not. Okay? Now, each of these different bases have specific names to them. One of them we've already talked about is called adenine. And we've already talked about adenine because, remember, that's the one that is on every ATP molecule. So an ATP molecule is just a nucleic acid that has 3 phosphate groups, but it has the specific adenine nitrogenous base. It's called ADP because it's adenosine triphosphate because of the adenine. Okay, the other ones are guanine, or I should say the other purine is guanine. So adenine and guanine are the double ring purines. And then cytosine and thymine are the single ring pyrimidines. And is that big enough for you guys to be able to spell? See that? Mostly? Bigger. Is that better? Okay. learn how to pronounce these: adenine, guanine, cytosine, thymine.
Those are the 4 nitrogenous bases.
Those are the 4 nitrogenous bases. Now, what happens in the DNA strand is you have this one chain of phosphate groups and sugars, and the nitrogenous base is hanging off the side. You've got another chain of the phosphates and sugars with the nitrogenous bases hanging off the side. When they come together, the way that these purines and pyrimides hook up, that gives you the rungs of the ladder, but the way they bond, that also gives you the twisting pattern that makes the DNA spiral around, causes that helical shape. Okay? Now the way it works is adenine always is attracted to thymine, and vice versa. You will not catch a adenine molecule hanging out with a guanine or a cytosine. What's that?
So thymine will never be without cytosine.
Right. They're exclusive to each other. They're in an exclusive relationship. Guanine and cytosine always bond together too. So normally we just refer to these by the first letter. So we say A goes with T and G goes with C. And that's the way it's going to be in every molecule, DNA molecule, throughout every life. Because of that specificity, that makes your cells able to read your genetic code. It makes your cells able to build new DNA molecules so that it can copy its DNA and chromosomes. But for a lot of different things that your DNA can do. Now, you may notice here in both cases we have a purine and a pyrimidine that are matching up. And because we have a double ring and a single ring structure that match up in every single case, that means that the DNA strands are going to be the same distance apart from each other. Because these, you take these and put them together, and you always get 3 rings across. So when we look at a strand of DNA, let's go back to that here, we can see that, these chains can stay the same distance across from each other because of the way these things form up. But it doesn't matter what side they go on. Here we have an A on this side and a T on this side. Here we have an A on this side and a T on this side. In this case, it kind of alternates with a and T and G and C, A and T and G and C. But that doesn't necessarily have to be. We could have 3 T's in a row on this side and 3 A's in a row on this side. And we'll learn how all that works when we start looking at how your genes are actually expressed. Okay? But it's really easy to figure out what's going to be, what the other side of the DNA molecule is going to look like, because if you can read one side, the other side just has to mirror it. If one side goes A, G, C, the other side has to go T, T, C, G. It always has to mirror what's going on the other side of the DNA molecule. Okay? Now, when we look how the sugar and the phosphate groups link up, they kind of have what we call directionality to them. So they're pointed in a certain direction. On one side, we can see here, it runs from what we say is 3' to 5'. And on the other side, it runs in the opposite direction, from 3' to 5', this way.
It's not always 3 and 5.
What's that?
It's not always 3 and 5.
Yeah. Now, those names are not that important. There's a reason why scientists call them that. But what you need to realize is these things are kind of, but each strand is running opposite to each other in opposite directions. It's kind of like this. So the term for that is, we say they run anti-parallel. Maybe not too close. So anti-parallel means they're parallel to each other, but they're running in opposite directions. And that's the basic structure of DNA. Now, when you build your DNA molecules for your project, which some of you might actually start on over Christmas, some of you. Yeah.
Is your son outside?
Those are the things that you're going to need to consider. You're going to have to show the sugar and the phosphate groups. You're going to have to show the bases. You're going to have to make the bases different from each other and show that A and T and G and C always go together. And you're going to have to show that they wind in a helix. All that stuff is laid out on your rubric. Make sure you look at that when you're building it. It's kind of amazing how many people sometimes seem to go, well, I'll just build a DNA molecule and don't ever actually look at the rubric and what it actually means. Make sure you follow that along. But
Use those things.
what's that? No.
No, it's not cheesy.
You don't have, I mean, I don't know that I have enough of those to make a DNA strand big enough. When we look at this right here, a single nucleic acid here, I mean, there's got to be 10 oxygen and at least 5 or 6 carbon, a bunch of nitrogen. I mean, one box of that is going to build like maybe one.
How many nucleic acids?
10 on each side.
So 20.
So that's not going to do yet. Okay, now you don't have to build it to show every single atom, but that's what those are. Those show every single atom. Those don't show them all.
I was gonna say, better start on every atom.
Yeah. No, you don't have to show every atom, but that's what those are built at. You wouldn't be able to use those to build it because it doesn't have the right bonding pattern to be able to put it together like that. Okay. So that's that. We're going to watch a video here that'll talk a little bit more about Rosalind Franklin. Okay, here is your question of the day. Your homework for tomorrow is to do 8.0. You have it in your packet of stuff that I handed out last week. So you get started on that. Go for it.